Our introduction is,
What are we comparing/researching? We are comparing what is eDNA used for, eDNA versus the traditional methods, upsides and downsides of eDNA and the traditional methods, and we're going to contact an expert in their field named Jory Harrison to get her opinion on these highly valued topics. Traditional methods for identifying organisms and ecosystems. For the traditional methods, we have visual, audio, call-in surveys, footprints, netting, and electric fishing. We are calling these methods traditional methods because they have been around for a long period of time.
eDNA is a type of DNA found in the environment. It can be used to track a lot of animals. It's DNA from animals, so animals will shed it. Humans actually shed 40,000 skin cells per minute.
Problem.
The advantages of DNA are organisms leave it behind in water, so humans can collect it without ever having to go in contact with the species. It offers vital clues about the well-being of the animals and the aquatic systems. It can measure climate change and endangered species. Sometimes eDNA can focus in on one specific animal, and when combined with more tools, improve the ability to assess status and habitat.
Advantages of eDNA. eDNA degrades in water in 7 to 21 days, so you have to act fast. Rapid climate change slows down the process of eDNA. Additionally, the field of eDNA is still young and rapidly developing, and not that much information has been discovered yet. New researchers are constantly contributing to the development of better technologies for eDNA.
Method is
Experimental part, preventing contamination. First steps of experiment are: Step 1, find at least 3 Tupperwares. Step 2, wash Tupperwares very well wearing gloves. Step 3, put isopropyl alcohol on paper towel. And step 4, rub paper towel on Tupperwares to make sure that there is no contamination.
our experimental part, taking the samples. Take 15 centimeters into the water and submerge the Tupperwares inside. Make sure to collect mostly sediments, rock, and mud, and we can leave some water. We did this at the edge of 3 different places along the creek, one upstream, one in the middle, and one downstream. We labeled them with their depth and where it was taken and put them in the freezer to be preserved for later means of research. Our experimental design. This on the picture you see is our final product that we will be aiming for. Our control variable is a banana. Our independent variable is our sample, mud or the banana, and our dependent variable is the extracted DNA.
Extracting DNA Part 1. Instructions. Step 1. Add water and electrolyte salt to the mixing bag. Step 2. Find a sample and add it to the bag. Step 3. Mash up the samples into a slurry by pressing the bag. Step 4. Add the lysis buffer, which breaks open the cells, to the samples. Mash and mix. Step 5. Place the filter in a clean cup. Extracting DNA Part 2. Step 6. Pour the sample mix into the filter. Step 7. Wait 5 minutes to let the liquid pass through the filter. Step 8. Add 9 milliliters of isopropyl alcohol to the tube provided. Step 9. Pour a small amount of liquid into the isopropyl alcohol. And step 10. Watch the DNA precipitate and float up to the surface.
Our data is
Observations we had. There were lots of clumps in the banana sample that we had, and a lot less clumps in the samples that we took from the creek. The banana sample worked better than our creek sample, and we also learned that mud is hard to get DNA extracted from. Comparing eDNA to the traditional methods. eDNA. Some of the advantages eDNA has is it's very good at accuracy, it has low cost, high sensitivity, good detection, and is very short in time. It only takes 2 to 4 hours. Disadvantages. It's only approved to research on a few species and can detect only the absence of species. It can't collect info on life stages, health, or population.
Advantages. It is approved to research more species and has been around longer. And traditional methods can also detect info on life stages, health, and population. It can also detect if species are endangered or not, but there are some disadvantages too. Its detection, it has very low detection, it needs more sampling events, it's more time consuming, low density detection, and a lot costlier.
Conclusion.
eDNA has been used worldwide for multiple things. eDNA has been used to track polar bears and other elusive Arctic wildlife. In Australia, it's even been used to monitor coral reefs. It has been used in several other places to track endangered species. And when we interviewed our expert, Jory Harrison, she told us something that we're taking away from our project. It's best used with other traditional methods.
Future. Climate change is a big problem, and it slows down the process of the traditional methods. eDNA improves the ability to identify organisms even with climate change rising. And the field of eDNA is quite new, and this eDNA technology will continue to evolve in the future.
Anything could happen.
Yeah,
Knowledge
it's
base.
knowledge.
We acknowledge Dr. Sawyers, our amazing mentor, Jory Harrison, the expert we interviewed, Miss Madison, our help with citations, and Dr. Miri, our help with editing. Thanks.